Thanks, Shelby, for the introduction, and thanks for the invitation. It's really great to be here to talk today about functional metagenomic sequencing, which enables us to do precise modulation of the enteric microbiome. I do have an introductory slide, so I'll just go through that again. So who am I, and why am I talking to you today? So I'm a principal scientist at DSM-Firmenich. I also have an associate professor position at Scotland's Rural College. I come from a background of 20 years of research in animal health and nutrition, formerly at the Institute for Animal Health, now the Pirbright Institute, probably most famous for releasing foot and mouth disease into the environment, but we don't talk about that. There I worked on Murex disease in chickens, Salmonella in chickens, and TB in cattle. I then moved to the Roslin Institute, most famous for cloning Dolly the sheep. There I was working on microbiome and methane in cattle, microbiome, health productivity in chickens, and genetics in the microbiome. So on the right of the slide there is Dana Stanley. That's not me. Dana can't be here today to give her presentation, but what I'm going to do is try and present her work. She's run 3 trials on what I'm going to talk about, so I'll present those later on. Dana is a professor at the Institute for Future Farming Systems at Central Queensland University, and what she works on is poultry health and nutrition, probiotics, microbiome, next-gen biotics, and pathogen control. So what am I going to cover today? I'm going to talk about the microbiome and why we think it's vital to understanding farm animal health and productivity. I'm going to spend a lot of time really describing the importance of data and why we think data is important and our strategy towards data. I'm going to talk about our dual strategy, which allows us to harness data to optimize food production and design next-gen feed additives. And then I'm going to present to you results from 3 trials which were run by Dana's group in Australia. In case anyone in the room doesn't know, what is the microbiome? So there's a definition there by Joshua Lederberg, the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. So basically, it's the bugs that live in and on us or live in and on an animal. So we've known about the microbiome for a very long time. I think this is the earliest publication I could find. I'm going to try and pronounce the name there. I think it's Leeuwenhoek. This is in 1684. Basically describing some microscopic observations on tooth plaque. And there, upon looking down the microscope, he describes very many small living animals which moved themselves very extravagantly. I think we'll all be doing that at the barbecue tomorrow night. So I think there's the first view into the oral microbiome in 1684. And if you go even further back, then Hippocrates famously said all disease begins in the gut. And I think we can be pretty certain that at that point he was talking about the microbiome. So why are we, DSM-Firmenich, why are we as scientists interested in the microbiome? So basically in any large animal, there are at least as many microbial cells as host cells, as animal cells. So it makes sense to treat these holistically. Often the microbiome is termed the second genome. So as an example, the chicken genome contains 30,000 genes that encode all the information that you need to build a chicken. The chicken microbiome, on the other hand, is estimated to contain over 20 million genes. A lot of functional diversity, a lot of information encoded in these genes and what they do. Both the host and the microbiome are under intense selection pressure, different types of selection pressure, but selection pressure nonetheless. And together, you need both the host and the microbiome to be optimized for healthy, productive animals. And finally, we feel very much that the gut is an interface between the diet, the environment, and the animal. If you think about a chicken, a lot of things are going in through the mouth, down the throat, and into the gut. And so we think and we see that the gut microbiome is the first thing to react to perturbations in diet, perturbations in environment, perturbations such as pathogens. So it's a good indicator of what's going on with the animals. But also, when you put something in there through the diet or you change the diet, those gut microbes have evolved key metabolism genes to deal with that. And we can see shifts in those genes when we change diets. So to summarize, the gut microbiome is, we believe, is a key driver and indicator of health and performance. Briefly, how do we study the microbiome? So we almost exclusively use whole genome shotgun metagenomics. We don't use 60S sequencing. So what that means is we take all of the cells in a microbiome, so all of the microbial cells in the gut, we extract all of the DNA simultaneously, fragment it and sequence it. This gives us millions and millions of sequence reads. These need to be compared to reference databases so that we can interpret them and know where they come from. We compare them to a genomics database to get microbial species abundance. We compare it to a pathway database to get gene and pathway abundance, and then we combine these to derive insights. Crucially, choice of that database. I've been a bioinformatician for 20 years, which means dealing with this type of sequencing data. Crucially, that choice of database is absolutely key. If you choose the wrong database at this point in your workflow, then everything downstream will be wrong, including lots of false positives and false negatives. So within the company, we've spent a lot of time optimizing our databases to make sure that they're really, really highly accurate. We started off really taking all of the publicly available microbial genomes. So these are sequenced microbial genomes that we know came from chicken guts, that have been cultured from chicken guts globally, that are in the public domain. We made a microbial database out of those genomes. And basically, when we mapped our metagenomic reads, those sequencing reads that come from the chicken gut, we were getting a mapping rate around 60 to 70%. So we're throwing away 30 to 40% of the data because we didn't know what it was. So we decided to fix that, and again, using a process that I don't have time today to describe, but if for any bioinformaticians in the room, we took publicly available data and our own data, and we assembled assembled genomes. We added those microbial additional genomes to the public data, and now we get upwards of 90% mapping rates, 93, 95% mapping rates, which means we're using all of the data that comes out of our omics assays of the microbiome. It gives us a better, more highly accurate estimation of the microbial community and the abundance, and it allows us to identify many more species that the publicly available data can't. And you can see, I'll just explain what the graph is. Basically, the graph is 5 different data sets. The grey bars show that 60 to 70% mapping rate from publicly available genomes, and the blue bars are our proprietary, and we think industry-leading database where we have 90 to 93% mapping rate. So we're very, very proud of that. Going back to this, the gut microbiome, we believe, is a key driver and indicator of health and performance. I want to give you an example of that. The example I'm going to use is CAZymes. So what are CAZymes? So CAZymes are, it's a shortcut for carbohydrate active enzymes. These are enzymes that are involved in the synthesis, metabolism, and recognition of complex carbohydrates, right? And they are absolutely abundant in the microbial genomes that live in farm animal guts. There are millions of these genes, okay? Driven by the diets that we put in there. Because, of course, we put a lot of complex carbohydrates into the diets of farm animals. So it's no surprise, really, that the microbiome contains millions of these genes. Perhaps surprisingly, and we'll talk about the graph in the bottom left there, you probably can't read the labels there. So basically in 2014, there was a paper published by Martin Sargent in PLOS ONE where he compared, one of the things they did in the paper was compare the composition of genes in different animal microbiomes. And one of the surprising results, if we think, for example, of ruminants, we think of ruminants as perhaps being these master degraders of complex carbohydrates, so the rumen will contain microbes with abundant enzymes to digest cellulose and hemicellulose and all these complex carbohydrates. But surprisingly, 1% of the genes in the rumen microbiome are devoted to carbohydrate metabolism. And it's actually higher in the chickens. 1.5% of what Martin Sargent found is 1.5% of the genes in the microbiome if the chicken gut microbiome are devoted to carbohydrate metabolism. And actually this has moved on since 2014, and 2 further publications, Segura Wang, who's one of our colleagues, estimated at over 2%, and Feng, who's just an independent paper in the literature, estimated that the CAZyme abundance in chicken microbiomes is above 3%. This is really, really high. If you think of all the things that microbes have to do, replication, defense, communication, structural genes, having 3% of your entire genome devoted to carbohydrate metabolism is really, really big. And it turns out, just looking at these CAZymes, forget about taxonomy abundance, forget about species abundance, forget about anything else. If we just look at CAZyme abundance, it tells us things about both diet and breed. If we look at the top right-hand graph, what that's showing there is basically if you take Ross 308 broilers and you feed them either a fish meal diet or a vegetarian diet, matched for nutrient content, so there should be no difference in the nutrient content, then basically the CAZyme abundance will separate out, will tell you the CAZyme abundance in the chicken microbiome will tell you which diet that is. You get complete separation on the PCA. This is a PCA of CAZyme abundance. The red dots at the top are, I think, the fish meal diet, and the green dots at the bottom are the vegetarian diet. So immediately, just one enzyme class, the abundance of the microbiome is telling us which diet those chickens have had. And then the bottom right-hand graph is a similar graph. This is basically the diet is fixed, and there's two different breeds. There's a Ross 308 and a Ranger Classic. And again, just the CAZyme abundance is telling you which chicken breed you're using. So this is it's just an example of how we can use the microbiome. The microbiome is telling us things. It's telling us about the diet. It's telling us about the breed. And what that means is there's information encoded in the microbiome that we can use for more useful things. And we already know the diet and we already know the breed, but I'm just giving this as an example of how the microbiome is an indicator of things that we're interested in. So DSM-Firmenich, we have this kind of three-prong approach to our innovation R&D platform. So at the top, the first prong, I think, is working with food producers and farmers, where we record health metrics, environmental data, performance metrics. Then we record a bunch of biomarkers, and we record microbiome information. I think Aaron will talk about this later on. And basically, the main goal of that work stream is to optimize food production for those food producers. But there's a secondary impact there, that over time what we do is we build up a database linking microbiome biomarkers with health and productivity outcomes. And what we can do then is use that in the second stream, which is our R&D platform, where we have a bunch of preclinical platforms, omics platforms, and obviously we run trials that then allow us to develop feed additives that address those microbiome biomarkers that we think are associated with productivity and health. And then we have a data science platform, which includes bioinformatics, machine learning, and AI tools. So our goal is to understand the beneficial function of microbes and encourage them to do more of those things. And as I said, we have this, there's a cartoon there of the kind of process of innovation, but it starts with understanding from food producers what makes a good and healthy, productive animal, productive chicken, and the microbiome biomarkers and host biomarkers that underpin that, designing feed additives that address those, through a bunch of preclinical screening and clinical trials. And together, putting that all together in a data science platform then enables us to develop new and innovative products. So enough about the kind of company strategy, if you like. One of the platforms we have for developing novel feed additives is our precision glycan platform. Briefly, what this does is we take simple sugars, monomers, and then we have a bunch of processing conditions. We call these recipes, and basically what they do is they turn the monomers into synthetic complex carbohydrates. And if we know that if we run the same recipe twice, it creates the same synthetic glycan, but we also know that if we change the processing conditions, if we change the recipe, it leads to different glycans. What this means is we can produce millions of different types of synthetic glycan. And this can be put into our screening platform to try and optimize particular parts of the microbiome, because they address different parts of the microbiome. So to convince you of this, here's a plot that we generated fairly recently. This is from one of our preclinical workflows. It's a very messy plot, but stick with me. What we have here is a PCA of species abundance, taxonomic abundance, and what we have on there is 14 different synthetic glycans. And what I'm encouraging you to see is that basically the data points cluster by glycan. So what we have is reproducible impacts and different precise reproducible impacts of the different synthetic glycans that we can create. And because that's a bit of a complex plot, I've pulled out a simpler one. What we have here is a control group, a glucose monomer, and just one of those synthetic glycans. It happens to be the precision biotech I'm going to be talking about today. And what you can see here is very clear clustering according to the input that we give to the chicken cecal microbiome. Reproducible taxonomic changes induced by these synthetic glycans that we can create. So let's move on to the trial results. This is normally where I'd hand over to Dana. Unfortunately, she can't be here today, so what I'm going to do is I'm going to try and present to you the results of 3 chicken trials that I wasn't involved in running. So please bear with me, and I'll try and get over the messages that Dana was trying to get over. So, first trial I want to talk to you about is a layer trial. These are layer hens from a free-range farm in Southern Downs in Queensland. So these are on-farm trials. This is not a research trial. It's an on-farm trial with all the mess that happens in real-world trials. 2 identical sheds, each shed with 40,000 hens. They have the same feed and the same water supply. We had a control group and a group with a precision biotic that we produce input into the feed at a rate of 900 grams per ton. Sampling at 28, 50, and 72 weeks. All performance parameters were recorded by the farm veterinarian for the duration of the trial. And this is just a kind of a picture of how the farm was set up. So exactly the same feed, exactly the same water supply. The only difference is the precision biotic in the right-hand shed and the control group in the left-hand shed. Those are the performance metrics measured. I'm not going to read them out, but you can see kind of mortality, rate of lay, grams of feed consumed per day, etc. And just to point out that this is a published trial, so if you want to go and read more about it, then it's been published in the Journal of Animal Physiology and Animal Nutrition. A lot of detail on this slide. So what I encourage you to look at first of all is the plot in the middle at the top, plot C. What this is plotting on the y-axis is hen house eggs, and it is expressed as the difference between the precision biotic group and the control group. So bars beneath the line show there's more eggs produced in the control group, and bars above the line show there's more eggs produced in the precision biotic group. You can see early on in the cycle there's slightly more eggs produced in the control group, but later on in the cycle there are far more eggs produced by the precision biotic group, and over time that's absolutely dominant, and we see a much bigger outcome in the precision biotic group in terms of the number of eggs produced. And then I'd like you to look at panel D, which is the bottom middle graph. This is, on the y-axis, is feed conversion ratio, again expressed as the difference between the precision biotic group and the control group. And what we're looking for there, ideally, is bars beneath the line showing that the FCR was lower in the precision biotic group. And here that tells a similar but even better story, showing that pretty much throughout the entire cycle, the precision biotic group had a better FCR than the control group. A slightly more complex story than I've led on so far. So if you're wondering what that blue box is, this is a smothering event. Again, these are on-farm trials, so this is not an R&D trial where we control absolutely every parameter. This is an on-farm trial. We know smothering events happen. They're unfortunate. This particular smothering event happened in both the control group and the precision biotic group. But I would like you to focus a little bit on, I'm going to try and highlight this, panel B. I'm not going to be able to see that. Hold on. So we have the smothering event here in blue. What we have is mortality, cumulative mortality on the y-axis there. And we have the green group, which is the control group, and the brown group, which is the precision biotic. And during the smothering event, I hope what you can see there is there is a lower mortality in the precision biotic group than there is compared to the control group. So there's some interaction there of our precision biotic with the smothering event, and we've got a reduced mortality in the precision biotic group. And then finally, and again, I do apologize, these aren't my slides. This is, again, a PCA of the taxonomic abundance from the microbiome. These big clusters really are clustering by age. Again, age is one of the biggest. It really dominates the abundance of microbes in the microbiome. So what we're seeing here is clustering by age, but actually within each age group, we see a shift which is expected in the precision biotic group compared to the control. So we are seeing that microbiome shift in these chickens. Dana's team also analysed RNA-seq throughout the gut. This is looking at host gene expression changes rather than the microbiome gene expression changes. So looking at host RNA-seq in different parts of the gut, input into pathway analysis for the differentially expressed genes, and what they showed was downregulation of genes in the precision biotic group, downregulation of genes associated with aflatoxin B1, downregulation of genes associated with LPS, downregulation of inflammatory response genes, and a significant reduction in the liver fibrosis pathway. these results, specifically in the liver fibrosis pathway, were backed up by some histology in the liver. So again, on the left, we've got the control liver. On the right, we have the precision biotic liver. And these black arrows are pointing to congestion of the liver blood vessels, which we see in the control group, but not in the precision biotic group. We'll come back to this later because we see a replication of the result in one of the other trials. So overall, trial 1, I think some really good results for our precision biotic, showing improved FCR, higher numbers of eggs, and some results from the host showing some really positive impacts. We'll move on to layer trial 2. This is, again, a slightly more complex story. This is unpublished so far, but will be published soon. The headline output of this is that comparing the control group to the precision biotic, in the precision biotic supplemented group, there were over 20,000 more eggs produced compared to the control group. Like I said, these are on-farm trials which have all of the problems associated with chicken production. Unfortunately, there were 3 disease outbreaks during this trial, 2 disease outbreaks of spotty liver and 1 disease outbreak of cholera. And I'd like to basically, again, look here on the left-hand side of the slide. This is the eggs produced over time. The green group is the control group, and the brown group is the precision biotic group. And you can see from basically from about week 30 onwards, there's just a much higher number of eggs produced in the precision biotic group compared to the control group. So some really nice results there. I think the thing that we need to think about and give a little bit more explanation to is that there was a slightly higher mortality in the precision biotic group compared to the control group. When I asked Dana about this, basically what she said was that because of the 3 disease outbreaks, there was a lot of very, very sick birds that weren't producing eggs in both groups, and those very, very sick birds died in the precision biotic group and didn't die in the control group. I don't currently have an explanation for that, but that is the observation that we have for it. But the headline figure remains that the egg production was much higher. We skip over that. And then the final trial I want to talk to you about is a broiler trial. This study was conducted using over 30,000 male Ross 308 broilers, 1 control shed and 2 precision biotic sheds. The 12 pen replicates with 900 birds randomly assigned. Again, unfortunately had a disease outbreak during this, infectious bronchitis on day 19 in the control shed, and about 10 days later in the precision biotic shed. This time, again, the mortality was switched, so you had a higher mortality in the control group compared to the precision biotic group. Again, this is published if you want to find more details about this. Looking at performance metrics in the broilers, headline figures are basically the precision biotic treated birds had a higher average body weight than the control group, and they also had a better FCR. So again, these are not my graphs, but basically what you have here is the body weight, and the blue bars of the body weight. You've got a control group showing a lower body weight than both of the precision biotic sheds. And then on the further y-axis here, that is FCR, and you can see here the control has a higher FCR than either of the precision biotic groups. So we're improving body weight and improving FCR. And then in mortality and EPF, the precision biotic treated birds recorded an average 17.5% lower mortality and a 106 points improvement in EPF. And again, we have these plots here. On the left-hand y-axis is the mortality, and you can see higher mortality in the control group than either of the precision biotic groups. And then on the right-hand side, we have EPEF, which is lower in the control group compared to the precision biotics. So again, quite really, really good results for the precision biotic there in the broiler trial. So in the broiler trial, there was also some interesting results from the liver. Again, you'll remember I mentioned histology in the liver in the first layer trial. Actually, these results were replicated in the broiler trial, where we have, again, that kind of blood vessel congestion in these dark red spots in the left-hand side in the liver that's just not present in the precision biotic, which gives us some confidence that the precision biotic is actually not only impacting the microbiome, but then also having a beneficial impact on the host as well, on the chicken itself. I asked Dana if she could summarize, because she sent me a lot of information and a lot of slides. I said, can you summarize this into some reproducible trends across the 3 trials? And this is what she said. So the precision biotic is reducing the abundance of E. coli, Galactobacterium, and true Clostridium. It's reducing the abundance of the Proteobacteria phylum. I know from my own research that often the Proteobacteria phylum is associated with inflammation in the gut. Also, is, as Dana says, home to some of the worst human and animal pathogens. So reducing the abundance of that phylum is good. Precision biotic reduces the number of species that have access to the ileum, reduces pathogens like Clostridium having access to epithelial cells. We get an increase in goblet cells, increase and better flow of blood flow to the liver, better disease resistance, and an improvement in gut health. And again, I would encourage you to go and read these papers by Dana Stanley's group if you want more information on this. A brief thank you from Dana. This is, I think, Dana there, and this is her research group. I want to finish with some conclusions. So I've given you a lot of information today and results from 3 different trials. I just want to iterate here that DSM Furniture are really convinced that we can leverage the power of data and understanding the microbiome to design innovative new products and services. We believe very strongly the microbiome is a key indicator and driver of performance and health. We work with food producers to monitor performance and health of the animals, but also measure host and microbiome biomarkers. This is a 2-for-1 win because it allows us to optimize the food production of those particular food producers, but it also allows us to understand the microbial biomarkers linked to performance and health. That knowledge enables us to design these new precise feed additives that go in and modulate very specific parts of the microbiome. One such feed additive I presented to you today, it's a precision biotic that shows, I think, quite impressive results in 2 layer trials and 1 broiler trial. 2 of those are published, 1 will be published soon. And it's just one example of how our glycan platform offers us the ability to precisely modulate the microbiome to optimize chicken performance and health. I'm going to end there and say thank you very much.